All right, we're going to talk about beer and anaerobic respiration. To understand anaerobic respiration, we need to remember what NADH does and what FADH2 does. So let's just look at glycolysis. Glycolysis breaks glucose down into a 3-carbon molecule called pyruvate, and electrons are released in that process. We stole electrons, right? We oxidized it. We oxidized pyruvate. And those electrons eventually go to the electron transport chain where they're used to reduce oxygen to water, right? Make sure you can use those words. Oxygen is reduced to water. It gains electrons, right? It's reduced to water. But what would happen if there wasn't much oxygen around? Well, if there's not enough oxygen around to pull electrons down the chain, everything from here on just stops. All of this stops. So you can still get a couple of ATP out of each glucose, but that's not enough to sustain a human. Like, you can't hold your breath and just run on that. But some smaller organisms can live off of that. And some of our cells do live off of that for a while, like muscles when we're exercising and things like that. But our whole bodies can't sustain life that way. So if there's not still oxygen around, which processes would still function? Well, glycolysis, and that's about it. So this happens when you're like lifting weights, but you don't have enough, I'm simulating that. You don't have enough oxygen in your blood to fully supply them with all the oxygen they need. Or I know one exercise that does this a lot, it's very anaerobic, is fencing, where you do short bursts of activity without getting your breathing up. So what happens in glycolysis is you're breaking glucose, which is 1, 2, 3, 4, 5, 6 of these carbon molecules. You're breaking it in half, and that releases electrons, right? And they get stuck to NADH, or NAD, which makes NADH, right? Now, this would go to the electron transport chain, but it can't because it's not functioning. There's no oxygen. But this is going to produce 2 total ATP, or 2 net ATP, right? 2 ATP are produced by this. Your body still, or the cell still wants more ATP, but it can't make any more until it gets rid of these electrons and frees up the NADH to grab some more electrons. So what's it going to do? Well, here's what it's going to do. We break glucose down into pyruvate, right? 2 pyruvates. NADH has these electrons, and it can't go to the transport chain, so instead it gives them back to pyruvate. And hydrogens come along as well. This hydrogen comes along, right? So let's look at what happens here. So in humans, we do what's known as lactic acid formation, or fermentation, right? Lactic acid fermentation. Fermentation, we're going to talk about fermentation. These are both forms of anaerobic respiration, meaning respiration without oxygen. So in glycolysis, we make 2 ATP. We've talked about that already. Glucose is broken down to pyruvate. There's pyruvate. 3 carbons, right? NADH is made, but it can't take its electrons to the transport chain. So instead, it gives them to pyruvate. So here comes pyruvate. Boom. These electrons are given back to it, and that makes this molecule, which is lactate. Lactate forms lactic acid when it's dissolved in water. So that can later on be converted back into sugar again, or broken down further when there's oxygen around. But for now, it's lactate. Lactate's also found in yogurt and milk products and things. So that's what happens in us when we do this. It's called lactic acid fermentation. It's a type of anaerobic, anaerobic respiration, right? Alcohol fermentation occurs in yeast and some bacteria. We don't do this. Otherwise, we'd all be really fit, right? What happens, it's almost the same as what we do. They do the same thing. Glucose is broken down to 2 pyruvates. Once again, there's your 2 pyruvates. But they have an extra step that we don't have. This molecule right here, that carboxyl group breaks off, forming carbon dioxide. So each pyruvate produces a carbon dioxide. We do that only if the transport chain's working. but they do it anyway. They release carbon dioxide. So their pyruvate is oxidized, and that produces acetyl aldehyde here. It's not acetyl-CoA because coenzyme A is not added, but it's just the 2-carbon acetylaldehyde. So there are 2 carbon molecules here. Now those have the electrons dumped back onto them, right? And you end up with this molecule, which is ethanol, ethyl alcohol, the stuff that you find in beer. So when you make beer or you make wine, the way you do it is you get a container. At least this is how I do it. And you fill it up with water, of course, and sugar of some kind. Now, if it's beer, it might be barley grains and things like that. It'd be sugars from grains, not just like table sugar. If it's wine, it'll be grape juice or sugars from grapes, right? So you end up with this sugary water full of yeast, and then you put an airlock on it. An airlock lets gases out, but it does not let gases in. Okay, so gases out. So that yeast in here is eating the sugars, and it's pooping out carbon dioxide, but no oxygen can get in. So pretty soon this yeast runs out of air, and it starts doing this process here, and it starts pooping out alcohol. So ethanol builds up in here. Then later on, what you can do is you take a bottle of beer and you fill it up with this liquid, maybe up to about here, a little higher. You put a little bit of sugar in here, and then you put a cap on it. And then the yeast eats that sugar. Hopefully you don't put too much sugar in there, because if you do, you'll blow up your bottle. But the yeast eats that sugar, and the CO2 can't get out now. So the CO2 stays inside, and it carbonates the beer. Now, if you are stuck at home in isolation, don't make beer unless you're 21 or over. But what you can do is you can look up a recipe for homemade ginger ale. And you can do a very similar thing. You can carbonate your own ginger ale using yeast, and a little ginger and some sugar and an empty 2-liter bottle. But be warned, if you don't put it in the fridge in time, that bottle will swell and swell and swell and possibly blow up. So something you might look into when you're at home. So, just a reminder, glucose comes into the mitochondria. Actually, that's a lie. Glucose comes into the cell. Here's the cytosol, right? Glycolysis takes place, breaking up that glucose into pyruvate. If no oxygen is around, fermentation kicks in, a type of anaerobic respiration. So fermentation. And that might make ethanol, or it might make lactate. Or it could be other things if it's some kind of bacteria doing something else, which we won't worry about. If oxygen is around, we have aerobic cellular respiration. And so that would mean that the pyruvate is broken down to acetyl coenzyme A, and then it will enter into the citric acid cycle. And that's the end of the food, right? The food is no more. It's breathed out as carbon dioxide gas. It's done. But the electrons from that food leave these processes and go to the ETC, which would be here in this, well, actually, we can't even see the inner membrane here, but it would be in the inner membrane here. Maybe right here. It's right here, I guess. Here's the inner membrane. They would go to the ETC, and then hydrogen ions would be pumped, and we would make ATP out of all this. And ATP is what powers everything you do. All right, that is cellular respiration. Now, I want to show you that this is a metabolic pathway. Glucose comes in, and there's a whole bunch of enzymes involved. This blue box up here is glycolysis, right? Pyruvate is produced, da-da-da-da-da. We've talked about this. There's an enzyme right here in glycolysis called phosphofructokinase. Phosphofructokinase is the funnest enzyme to say. And I wanted to point out that citrate, which is that first product of this cycle right here, of the citric acid cycle, citrate actually inhibits phosphofructokinase, either competitively or non-competitively, I'm not sure. It actually slows it down. And ATP also slows this down. So if you have tons of citrate being produced or tons of ATP being produced, it will slow down respiration so that you don't overproduce ATP. Because ATP can't be stored for long. It's used pretty much immediately after it's made. You don't have a storage unit for ATP. You store energy in the forms of fat or protein, and then you turn that into ATP essentially when you need it. So phosphofructokinase will slow it down. And then another molecule called AMP. AMP is adenosine monophosphate. It's ATP missing 2 phosphates. If there's a bunch of AMP around, it actually stimulates phosphofructokinase and gets it to go faster. So that's kind of interesting. So that goes back to those ideas of feedback, negative feedback mechanisms. I talked about this before, but let me reemphasize that. Negative feedback is how we maintain homeostasis. Negative feedback is not bad. Negative means that it's in the opposite direction of the stimulus. So in this case, we have way too much ATP, so we're going to move in the opposite direction. We're going to slow down the production of ATP, right? Just like if you have way too much blood sugar in your blood, you're going to react by lowering that blood sugar. That's what negative feedback is, and that's how homeostasis usually works. And so this is an example of that. Too much ATP around, it slows down the enzymes in the process. It stops the production of ATP for a while. All right, that's it for this lecture. We're going to move on to photosynthesis, and I recommend that you work on concept maps for this chapter, work on the guided reading questions, make sure you can kind of map out these processes, understand what oxygen is for, how hydrogen ions are involved in this process, how electrons are involved in this process. This is going to take a little bit of studying to grasp it and get a hold of it, but you can do it, and it's very important that you do so. All right, see you soon.